Over the last 200 years, human life expectancy has climbed steadily, but our health span, the actual number of years we live completely free from chronic disease, is lagging behind. We are living longer, but we are spending more of those extra years sick. A group of researchers in the Netherlands noticed a stark exception to this rule. They've been tracking participants in the Leiden Longevity Study, entire families who managed to stay highly active and disease-free deep into their twilight years. This chart shows the average onset age of chronic illnesses in the general population, compared directly to the offspring of those long-lived families. On average, these individuals develop cardiometabolic diseases a full 13 years later than their own partners. Wealth, environment, and diet play huge roles in how well an individual ages. But those external factors cannot explain a consistent 13-year generational advantage that appears regardless of a person's specific lifestyle. The resistance to disease was being passed down from parent to child as a heritable genetic trait. For the researchers, it signaled a race to find the specific biological code responsible for this delay in aging. Across 200 sibling groups, the team filtered 20,000 human genes down to 4 specific regions, isolating 350 suspects. Discarding genes that didn't alter protein function shrank the list to just 12 rare genetic anomalies. Among those 12, they found a mutation on the cGAS gene, short for cyclic GMP-AMP synthase. This was an unexpected find. cGAS was previously understood only as an emergency alarm for the immune system, but its presence in these families suggested it played an entirely different role in the biology of aging. Normally, cGAS acts as a cellular tripwire. It detects DNA that has leaked out of place due to viral infections or cellular damage. When it finds that misplaced DNA, it triggers an aggressive inflammatory response to clear the threat. While that inflammation is necessary for immediate defense, its long-term cost is high. When this pathway remains active over decades, it begins to damage healthy tissue, functioning as a primary driver of the aging process. There's a dangerous trade-off. Completely shutting the pathway down leaves the body defenseless, but keeping it fully active destroys tissue. The long-lived families possess a genetic bypass, born with one active cGAS gene, cutting their inflammatory output in half. This half-powered state places them in a Goldilocks zone. It produces just enough inflammation to survive infections, but not enough to cause the widespread tissue degradation that defines old age. While the correlation in these families is striking, scientists still need to prove that the mutation is the specific cause of their longer health span. To confirm this, researchers at the Max Planck Institute are now introducing this exact cGAS mutation into a living model, the African Killifish. This fish is the shortest-lived vertebrate known to science, with a natural lifespan of only 3 to 9 months. Using them allows the team to observe the effects of the mutation across an entire lifetime in a fraction of the time it would take humans. By comparing these fish to a control group, the researchers can verify if dialing down this single pathway measurably increases both lifespan and tissue health. If the killifish thrive, the stakes for human medicine shift. It suggests that rather than treating the various diseases of old age individually, we might eventually extend health span for everyone by artificially replicating this genetic glitch.